What I'm going to talk about now is some of the slightly more clinical research that we're doing in the group in Nottingham. And in actual fact, all these things translate together. So, for example, the cathepsin K project, then when we recruit people to our studies, we can actually use those people to look at levels of cathepsin K in their blood and tie in with the laboratory research we do. And so one of the nice things about having the LAM Centre and having people come and having people interested in research is that we can use the laboratory resources and the clinical resources, and that makes the centre quite powerful, I think. Anyway, so I'm going to talk to you about this project particularly because I really just wanted to say thank you for all the people who participated in this particular study. So although it's not completely finished piece of work yet, I wanted to sort of show what your generous donations of all those blood samples that I take from people in clinic is doing, really. And this study is really to answer the question of what makes LAM such a variable disease, and how can we predict which path people with LAM are going to go down? So this thing here is usually termed a spaghetti plot for obvious reasons. Each one of these lines, each piece of spaghetti is a person, and it's their FEV1, their lung function value, that's the lung function value that's important in LAM, over time, some of these people for a long time. And what you notice is that LAM's a really variable disease. So I've picked out 3 patients here. The person in green, who I've looked after for quite a long time, whose lung function's high, it's in the normal range and has been for many years. She's completely well, she's had no treatment. Then we have perhaps this sort of average, or what people used to think of as a typical patient with LAM, in the blue line here, who loses lung function at an accelerated rate but not super quickly. And then we've got a small number of people like this person highlighted in red here who run a much more difficult course. And we don't really know why some people behave like the person with the green line and some people behave like the person with the red line. If we knew, we might be able to turn some of those red people into green people, which would be the thing to do. And also, of course, we'd be able to advise people when we saw them initially how things were going to go. And we could reassure them, or we could target them for treatment much earlier, so we could hopefully prevent lung destruction. The difficulty we have is that lung function trajectory, whether you're a green, a blue or a red, is really difficult to predict over the short term. You've probably seen this slide before, but this is the FEV1 of someone with LAM over about 12 years or so. And each one of these dots is an FEV1 measurement. And actually, if you look over short periods of time, you get sort of different results. So in this point, if you draw a line through these, it almost looks like the person is getting a little bit better. If you look at this bit in isolation, they're obviously doing a reasonable amount worse. And then this bit at the end here is sort of leveled off. But actually nothing had changed much over the course of time here. No treatment or anything. This was from a long time ago, pre-rapamycin days. Actually, really, if you look at overall, it's a sort of steady change. But if you look over short periods, you get the wrong idea. And so it's quite hard when you've only seen somebody for 6 months or even a year or even 2 years sometimes to tell exactly what's going on and what the longer term picture is going to be. So some clinical features predict outcome. Earlier age at onset, just for having LAM for longer, is reasonably self-evident. If your first symptom's breathlessness rather than, say, pneumothorax or a kidney tumour, then that's probably not such a good sign. If you have weight loss when your disease starts, that's not good. And if you have this protein in your blood that's called VEGF-D that's elevated, the higher that is, probably the more active your disease is likely to be. And this blood test that can be used for diagnosis of LAM is also the only biomarker we have to predict the future with LAM at the moment. So we really need more things like VEGF-D to try and predict what's going to happen in the future. And if we had these things, we'd be able to advise people much more accurately about their prognosis when we saw them straight away. We'd be able to target people early with drug treatment. So rather than thinking, well, I don't know whether to put you on rapamycin and sort of thinking about the whole business for a year or longer because we're not sure what the lung function's doing over this short period, we'd be able to just think, right, well, this person isn't going to do so well, so we're going to offer them treatment today. We can also improve the design of clinical studies. One of the difficulties with LAM is that because it's variable, when we put a lot of people in a trial, some people stay well and some people don't stay well. If we knew that we wanted to try something where people might have a change in lung function over time, we could test which ones those were going to be, and then we could make the trial much shorter, much smaller, much cheaper, and we could test things much more quickly and easily, which would obviously bring benefits in research. And if we understand biomarkers, we know a bit more about the disease, and perhaps we can target things properly. So to answer some of those questions, we've started this project, which is now called the BioLAM study, which wasn't my idea, by the way. And really this aims to understand what are the factors that might predict the future clinical course of LAM. And we're looking at clinical factors, so did you come with breathlessness or pneumothorax or kidney problems? What do the features of the lung biopsy look like? Is there anything about the lung biopsy that's going to predict the future course? Are there blood tests? Is there anything in the genetic makeup of people? And the study was funded for 2 years by the Rare Disease Translational Research Consortium, who are part of the National Institute for Health Research, runs through the LAM Centre. And this is Suzanne Miller, who was the postdoc that we funded to run the study. So, as I say, we're looking at a few different things, and we're getting other experts to help us out. For the clinical data, we've got all manner of information about lung function at presentation and about different aspects of the disease phenotype, which we're analysing. We're using a statistician, Tricia McKeever, to help us with that, because you can actually do some quite powerful techniques with this amount of data. We're looking at pathology, and we're collaborating with a respiratory pathologist in Nottingham, Urshan Sumro, to look at the pathological features. And we've got an expert in analysing big genetic studies, Louise Wayne, who works in Leicester, helping us with the genetic side of things. So actually, we just heard this week that we got funded through another local Nottingham scheme to get somebody with expertise in mathematics to put all this data together to see if we can use all these different factors with large numbers and data sets to predict the outcome in the future. So the aims really are of the thing I'm going to talk to you about is to use different serum proteins to try and identify things that are abnormal in LAM, to identify things that are associated with how severe the LAM is, and maybe understand some of the pathogenesis. So I'm going to just tell you about one strand of this study, really, as an illustration of what we're up to. So the people that have participated are anybody that comes within about 50 yards of me, really. So the study subjects have LAM by ERS criteria. I mean, it's been fantastic. I have not asked a single person to participate who has said no. I mean, I've never done any research like that before. We're up to 160 something, and that's 160 straight yeses. I mean, it's just remarkable, and I'm very grateful to everybody that's participated. So the study is fairly straightforward. They sign a consent form. And as part of their clinical care, we examine them. They have X-rays, CT scans, lung function, etc. And we also take blood, a reasonable amount of blood, and we measure some proteins and some DNA and some things. 12 months later, we see what's happened to them, and we look at their lung function again, and we see what's happened in the interim. So it's a fairly straightforward sort of study. So this particular study, we used a technique called proteomics to try and understand which proteins in the blood are abnormal in women with LAM. And you can do that in two ways. You can either guess which proteins you think might be abnormal and measure them directly using a specific method, or you can do what's called an unbiased screen, where you do a technique where you analyse all proteins. We used a technique called proteomics, which smashes all proteins into little pieces, analyses the pieces, puts them back together again, and sees which ones are more common than the others. I don't really understand it, but it's very good. So we analysed the blood from 50 women with LAM who'd never had rapamycin treatment, 20 control women, and we then took some more women with LAM and confirmed the results in a separate population, which is how it usually works. And so, yes, we use this mass spectrometry proteomic thing, and we collaborated with Nottingham Trent University. This is them doing protein. I think it works like this. There's a robot, and anyway, data comes up. And so what you end up with is a list of proteins, and we identified 126 proteins from our screen. There are more proteins in blood, but these are the ones we identified. And there were lots of common proteins: albumin, acid glycoprotein, immunoglobulins. You expect these things in blood. The interesting question is what's different between the women without LAM and the women with LAM. And actually, we focused on 2 proteins. We found that this thing, vitamin D binding protein, which it won't surprise you is a protein that binds vitamin D, was over 3 times lower in women with LAM compared with women without LAM. Vitamin D binding protein, apart from binding vitamin D, has various anti-inflammatory and acute phase effects. It controls the activation of inflammatory cells and does a lot of things other than simply binding vitamin D. But that is one of its main functions. And then we found this other protein, alpha-1 acid glycoprotein, that was actually a bit higher in LAM. And this is another acute phase serum protein, which is associated with the transport of drugs and also hormones around the body. So, for example, it binds the female hormones and transports them around in the blood. So these 2 proteins were abnormal in women with LAM. One was lower and one was higher. So the next thing you have to do when you've done something by proteomics is that you have to prove this by measuring the actual protein level using a specific test which is more accurate and more quantitative. And so we did this. We measured the serum proteins using a test called an ELISA, and the vitamin D binding protein was indeed lower in women with LAM, about 2/3 of the value of healthy women. And we did 2 groups of women with LAM, and it was the same in both of them compared with the controls. And we'd also looked at this alpha-1 acid glycoprotein, and that was still higher in LAM in the 2 groups of women compared with the controls. So the results were correct. And I'm going to focus mostly on the vitamin D binding protein because that's the one that's turning out slightly more interesting. So the next question was, okay, so vitamin D binding protein's lower in LAM. So if it was important, maybe it would be lower still in the people who had difficult LAM compared with the people who had milder LAM, for example. And so if you break up the group of women whose vitamin D binding protein we measured into people with more active disease and less active disease, it is in fact the people with a more aggressive form of LAM who have the lower levels of the protein in their blood. And if you compare the level of vitamin D binding protein with a measure of lung function, in this case, this is the gas transfer test, the DLCO, the one where you hold your breath for 10 seconds. You'll see that the people with you can see from the line, this regression line, that the lower values of vitamin D binding protein are associated with the lower values of DLCO. So we think that the patients is lower in women with LAM, and the people with worse LAM have lower levels of vitamin D binding protein. So we next asked, well, okay, so if low vitamin D binding protein is associated with severe LAM, does that mean the outcome's going to be not so good in the future? And this study's ongoing at the moment. But what I can tell you is, if you look at VEGF-D, the only known biomarker for LAM, that goes up when you have more active disease. You can see that if we take the people who we saw at year 1 and then we say, okay, so in a year's time, how many of them have needed treatment with rapamycin for their LAM? If you look at the VEGF-D level, that was significantly higher. If you look at the vitamin D binding protein, remember low is bad in this case, that was lower in the people who were treated for rapamycin, not significantly to the same degree, but actually the numbers aren't very big yet and we're accruing more data all the time. But it looks like there might be some mileage in vitamin D binding protein as a marker of active disease and possibly of outcome. We're correlating this with lung function, and you can see all of these graphs. They all move in the right direction, but we don't have enough data yet to say lung function a year later can be predicted by vitamin D binding protein. But as we learn more about it, then we hopefully will be able to do that. We don't think that the protein comes from LAM cells. We think it's a naturally varying thing in everybody, actually. And actually, if you happen to get LAM and you happen to have low vitamin D binding protein, you might do a bit worse. And consistent with that, if we suppress the disease with rapamycin, it doesn't affect the level of vitamin D binding protein, and it's not really correlated with VEGF-D, this other marker of how many LAM cells you've got. So I think it's an independent thing of LAM, but it's an interaction between your genetic background and having the disease. So the next question then, okay, so what about the genetic background? And proteins are made by genes, and everybody has random variation in their genes. They're called single nucleotide polymorphisms, or SNPs. And they use this random variation to see if X is related to Y, or somebody might get disease, or where your ancestors came from, these sorts of things. And individual polymorphisms in individual genes sometimes affect protein levels. So what we did was we genotyped women with LAM for specific polymorphisms in a vitamin D binding protein. And this one here, RS7041, just rolls off the tongue, doesn't it? So this particular change in the gene was associated with variation in the level. And if you've got, everybody has 2 copies of all their genes. If you've got 2 of these G-type genes, then you have a lot of vitamin D binding protein. 2 Ts, you've got much less. And if you're in the middle, your serum level's in the middle. So we were thinking, maybe we could use this genotype as a prediction for how LAM might run in the future. The other thing that vitamin D binding protein does is it's related to your level of vitamin D. So we thought we'd measure people's vitamin D levels, and we've only just started doing this. Now, in fact, on Thursday, I was just plotting these graphs, thinking, nearly LAM meeting time, and better hurry up. So we haven't done the whole study, but we've looked at vitamin D levels in 28 patients, and actually 5 or 18% of them had vitamin D levels below the recommended amount for the amount of vitamin D that you need. And actually, the lower vitamin D levels tended to have lower FEV1 and higher VEGF-D levels, that's the right way around, suggesting it is probably associated with the severity of LAM. There was no clear association with outcome, but this is super preliminary data, and we're working on that now. And so we're trying to characterize the whole data set and see how the vitamin D level correlates with the genotype and the protein level that we've also shown. So that's the study, really. So what does all this mean for LAM? Well, actually, if you look at vitamin D binding protein in other lung diseases like COPD, the smoking-related emphysema, or in asthma, or even in tuberculosis, you get more lung damage if your vitamin D binding protein is lower in those diseases too. So it might be a general thing that protects the lungs against damage in some way, and LAM might be consistent with these things. What we did see is that low vitamin D binding protein in LAM is associated with the presence of LAM and the severity of it. And actually, the gene polymorphisms can predict the protein level. So we can't yet predict LAM outcome from this, but our next move really is to do a bigger population with more outcome data, and we have the samples to do that from the NIH cohort, and we're about to start working through those. And hopefully a bigger sample set with more outcome data might give us a clearer answer. So we're quite interested now in the vitamin D axis and vitamin D binding protein in LAM, and can you use that to protect against advanced lung disease? These are very preliminary results, so I will thank you not to go on Twitter and say vitamin D is good for LAM, because we don't know that yet. And particularly, don't start taking vitamin D just because of this talk. If you take vitamin D because you need it anyway, that's fine. But these are just preliminary data I want to share with you, really just to say thank you for participating. I'd also like to thank the people who we work with, Tricia, Urshad, Louise and David, who are our collaborators in the various strands of this project, the people who funded it, LAM Action, the NIHR, the LAM Foundation funded the proteomics study, the Nottingham Pathology Node fund quite a lot of our LAM research also, and also, yes, thank you very much to everybody that contributed samples. Thanks.
I think in the past there's been some mention of a connection between LAM and osteoporosis.
Yeah, good question.
Vitamin D comes into that, possibly?
Vitamin D absolutely does. So two things that we're working on come into that. So the thing that eats your bones when you have osteoporosis is cathepsin K, which is also, Debbie's just told you about. It's also an estrogen-dependent phenomenon. And so there's a lot of parallels. And vitamin D also protects your bones against osteoporosis. You're absolutely right. These slightly seemingly odd coincidences probably reflect that some of the same molecular pathways are abnormal in LAM cells as are in the osteoclast, the cells that turn bone over. In fact, and vitamin D is actually important in a reasonable number of things now. We know that protection from infections, including respiratory infections, vitamin D is required for that. Yes, it's sort of becoming the sort of vitamin of the moment, really. But what's interesting was we didn't think, oh yeah, maybe LAM's, important. Maybe LAM's related to vitamin D. We found an entirely independent way by using the proteomics, which, to my mind, strengthens the association, I think. Barbara, yeah.
Yes, sorry. As the sun doesn't shine a lot in the UK, and it does in Africa, are people in Africa getting less LAM than we are here?
Well, we don't know the answer to that. So what we've shown so far is that vitamin D binding protein might be protective against lung damage in LAM like it is in some other diseases. So vitamin D levels are entirely dependent upon, are at least partly dependent upon sunlight. And so you would imagine areas with poor sunlight may do worse if it's vitamin D that's important, not vitamin D binding protein. So as I say, we're a little way off being able to answer all of these questions at the moment. But I'm hoping that we'll be able to firm this up in the next 6 months or so. Obviously, LAM in different countries also depends on how common the genetic abnormality that predisposes people to LAM is, and there's a lot of other things. As far as we know, nearly all races seem to get LAM, and we don't know of any race that gets LAM more frequently or less frequently at the moment. It seems to be a fairly widespread phenomenon at roughly the same incidence or prevalence, I should say.